This is the final project for Bio 181 at CSU Global, and the enzyme I chose is Catalase. To begin, we'll discuss the enzyme name, the organism, and the subcellular location of the enzyme. So Catalase is an essential enzyme found in the organism Bos bovine. It plays a vital role in protecting cells from the harmful effects of reactive oxygen species by converting hydrogen peroxide into water and oxygen. Catalase is primarily located within the peroxisomes of the cell in bovine. Peroxisomes are membrane-bound organelles found in eukaryotic cells. Next, we'll discuss the primary structure. The primary structure of the enzyme catalase consists of a sequence of 527 amino acids. The primary structure refers to the linear arrangement of amino acids in the protein chain. The primary structure of catalase provides the basis for the formation of secondary structures such as alpha helices and beta sheets. Next is the tertiary structure, or the secondary structure. These secondary structures are stabilized by hydrogen bonds between the amino acids in the primary sequence. This enzyme contains turns, helices, and beta strands, which you can see below in the diagram. Next is the tertiary structure. The primary structure of catalase ultimately folds into a three-dimensional shape known as the tertiary structure, which is observed in the photo on the screen. The arrangement of the amino acid chain in the tertiary structure is influenced by interactions such as hydrophobic interactions, disulfide bonds, and electrostatic interactions. Catalase pathway. Catalase in Bos peroxisomes plays a crucial role in maintaining cellular integrity and protecting against oxidative damage by efficiently breaking down hydrogen peroxide into water and oxygen. Additionally, it contributes to the detoxification of harmful compounds, highlighting its significance in cellular homeostasis and defense mechanisms. Next, we'll talk about the functional parameters for activity. Understanding these functional parameters helps in characterizing the activity of catalase and studying its regulation, kinetics, and potential variations. It allows researchers to explore the enzyme's role in different physiological and pathological conditions and develop strategies to modulate its activity if needed. So going off the slide, there's a bunch of different categories or key factors. So there's substrate specificity, optimal pH, which for catalase this would be between 7 to 8, and that's slightly alkaline. There's temperature sensitivity, which is optimal for catalase at around 35 to 45 degrees Celsius. There's cofactors and coenzymes. There's enzyme concentration. So if there's a higher concentration of catalase, this can lead to increased catalytic activity because it's optimal conditions. There's inhibition and regulation. So the catalase activity can be regulated by various factors such as inhibitors and activators. And finally, there's genetic variance, which may affect the efficiency of catalase. Okay, now we'll talk about chromosomal location. So to start, I'll give you a description of the gene, which is noted as CAT. Molecular structure. The CAT gene is composed of a specific sequence of nucleotides. It contains the necessary information to synthesize the catalase enzyme during protein synthesis. The gene serves as a template for transcription, where a complementary RNA molecule is synthesized based on its nucleotide sequence. This RNA molecule, known as mRNA or messenger RNA, carries the genetic code from the gene to the ribosomes for protein translation. The mRNA transcribed from the CAT gene is subsequently translated into the primary structure of the catalase enzyme. Catalase gene is located on chromosome number 11 on the long arm in the 13 cytogenic band. And you can see that in the picture right here. Next up is the complete gene sequence. So I couldn't fit it all on one slide, so over the next 3 slides is the complete gene sequence from 1 all the way until 1981 line. Following the complete gene sequence is the complete protein sequence, which you can observe here on the screen. And these are all the references that I used throughout the project. I was able to bring everything over, do a little bit more research, and get my thoughts together for this presentation. Thanks for a great class.